I was freezing. I still am. I know. They're actually really good, but it's prickly, that's why. Really, everything it's probably cold, it's sitting on my iPad. Well, no, I okay, it says you need permission. Brendan, how would I understand? I have 20 pages.
All right, so let's go ahead and get going today. Thanks for getting your exam resubmissions in. And so we're going to start building on where we left off last time. And so I've been kind of hinting at this particular topic for a number of lectures. And I think after seeing some good examples, now is a good time to kind of formally discuss this because it has implications for what we're going to talk about. I think in one of next week's lectures, when we start talking about basically like what vaccination is doing, and then also some topics in epidemiology. And so that's kind of where we're going to get through by the end of next week, and that will be kind of wrapping up more or less the sort of lecture, big lecture components of the course, but one more paper we're going to go through. And this evening or tomorrow morning, I'm going to be posting the third and final homework, help you prepare for the third exam, which is in a couple weeks, right? Is that right? 2 weeks? 2 weeks? 2. So 2 weeks. All right. So, this last kind of section of the class has been applied, and hopefully this is, giving you some interesting things to think about with your respective pathogens. There will be a final exam. Again, your lowest, should you take the final exam, well, regardless, your lowest exam score of 3 exam scores is dropped. That counts for 60% of your grade. Homework, you've been doing it. Been working through grading the second homework. Looks like you guys are doing well. Yeah, so just kind of take it home with the presentation, and you guys recognize many of your faces here in class. So I think by and large, everybody's doing pretty well. So we're going to talk about this particular topic today, the evolution of bacterial pathogens. So first of all, I think it's important to address this. Hopefully you've all seen it. How many of you guys have taken evolution here at Unzel? Awesome, lots of you guys. All right. So if you haven't taken it and you're a biology undergraduate, hopefully you're taking it next semester, because my understanding it's required to graduate. So I'm going to talk about some of these major mechanisms, at least in the context of pathogens, what's important. And we'll kind of sort of resurrect a few of the topics that we talked about in the last couple of lectures in a slightly different context in terms of what's driving the evolution of pathogenic traits. And then we'll kind of look at some applied kind of ideas in terms of what are signatures that we see in pathogens when we're out out in the real world, isolating these things, and what are some implications for this, and that's really what we're going to be getting into. So, biological evolution, despite all of the nuances and everything else, the cultural associations with this particular term, evolution, is simply the change in frequency of allele variance in a population through time. So this is a very strikingly simple concept that has very complex outputs. As we see, because we're all sitting here cognitively thinking, typing, listening to me, or not listening to me, or we're doing all sorts of really complicated things. We don't think they're complicated, but in terms of biological function, they're extraordinarily complicated. And so I often like to address terminology when we're talking about things. And these are words that have come up throughout this course. So if I'm talking about fitness, the context of evolution, I'm talking about the relative reproduction rate of a species in a population. And so this can be a population, a mixed population of microorganisms. So we're talking about E. coli's competition with 14 other species in your colon. Or we can think about this at sort of an intraspecific species in terms of a particular mutant in a particular gene competing against all of its other siblings. Okay. When we talk about phenotype, this is the observable behavior or function of an organism. And so, in the context of this course, it's been talking about things like metabolic traits or the ability to cause disease is a phenotype. And so, genotype, this is the genetic sequence variant of a particular species in a population. Okay? So, these two things are integrally coupled through the central dogma, right? So genes are replicated, transcribed, and translated into proteins that perform some particular function. And one of the grand biological challenges, I think, across all organisms is how well we can map genotype to phenotype, right? So that's one of the grand challenges in human genetics. So the extraordinary diversity that we see even in this room, what genotypic changes lead to these wonderful phenotypic changes we see in our own human population. And when we talk about mutations, these are alterations in DNA sequence leading to variation in genotype. So, there are a lot of different factors that shape evolution, such as mutation rate, population size, mode of reproduction, the number of species in an environment, and the number of species-species interactions in an environment, right? So, these are sort of various kind of straightforward factors. These are some factors that really get into sort of how does this particular gene interact with the environment, for example, or how are organisms interacting with each other in an environment. And so these really kind of where evolution and ecology are really kind of deeply intertwined. But I'm going to take you through some of these processes, kind of bolded ones, with the focus, one, on prokaryotes, and two, on sort of its relationship to pathogenesis. So, mutation rate. So this is basically how frequently mutations are showing up in a given population per site of the genome per generation. So mutations, the base, the replication event. And I always like numbers because it gives some perspective on this. So one of the crazy ones that I like to think about is that you have a bacterial genome. And so we'll say this is a big bacterial genome. And there's around 10 to the 7 base pairs. So this means there's 10 million nucleotide base pairs in this genome, which is, for bacteria, it's pretty large. And that there's an intrinsic mutation rate of around 10 to the minus 10, which leads to around 10 to the minus 3 mutations per replication event of this gene. Okay? So if I take 5 mils of LB and I assume that this organism grows up to around 10 to the 10 cells, total cells in a culture, and in the final generation leading up to this number, there's around 10 to the 10 replication events that happen. What we can back up from sort of crunching numbers, if we know that there's this many replications, there are this many mutations per replication, is in this standing population, there's about 10 to the 7 mutations. And so ultimately what this gets at is that nearly every non-lethal site in this bacteria's genome, so roughly about 30% of a bacteria's genome is essential. And so essentially every non-lethal site in the bacteria's genome has been sampled with some kind of mutation in this population. And what's interesting is that we actually don't really ever see this level of diversity in the lab. And an interesting question then, why? So this is the intrinsic mutation rate. Why don't we see mutations all the time? And one of the factors that drives this are some processes that we're going to get to later. One of them is population size. So population size is the number of individuals of a species in a given environment. And so I hope this is straightforward so far. And what ultimately, what we saw from our last example, is that population size dictates how many mutations occur in a population per generation. So if there's a given mutation rate, the larger your population, the more mutations that population is going to sample. Okay? And so what that means is that population size dictates a lot about the process of evolution. So if we go back to our definition, it's the change in frequency of a particular allele in a population through time. And so you can imagine if a population size is small, that when mutations arise, they might arise less frequently than in a large population. But by virtue of the population being small, if that mutation has some small benefit, or just it has no benefit, that it just showed up and is moving through this population just at some neutral sort of probability, it could be lost, it could increase in frequency, is that small populations, basically mutations can show up and quickly fix in a population. So when I say fix, when we're talking about this in terms of population genetics, this means that a mutation arose and its frequency increased from essentially nothing, 0.0001% of the population, to 100% of the population. So if a mutation exists in the entirety of a population, we now say that that mutation is fixed in that population. So that there's really no chance that that mutation can be lost. And so, because of population sizes being small, it's sort of the idea that, well, if there were 10 questions on a 100-point exam, and I said each question is worth 10 points, and I said either you get the whole question right or you get the whole question wrong, you're talking about huge, quick steps towards or away from 100%, right? So if I said there were 100 questions, and each one was worth one point apiece, and you're either right or you're wrong, that getting from sort of 100% right to 100% wrong takes many more events, many more steps to get there versus a small population. A large population, mutation can arise. It can kind of wander around. If it's neutral, it can be beneficial. And if it's not sufficiently beneficial, to quickly rise to very high frequency in the population. This means that it is more difficult for mutations to fix in large populations as quickly as they do in small populations. Now, mutations are more abundant, but competition for sort of benefit of mutation in large population sizes is much larger than it is in small populations. So, what do you think? What regime here do you think pathogens are operating? Do you think there's just a few of them, or do you think there's a lot of them?
A lot.
So we've got one for a lot. It could be both, right? So it depends on the context, right? So if we think about the story of salmonella, when you ingest salmonella in sort of contaminated water, there might be 1,000, 10,000 bacteria in that sort of a half liter of water that you drank. And under those conditions, we're talking about very small population sizes. So if there was one mutant in those 10,000 bacteria that you consumed that does better, that in the selective environment that is your body, that is quickly going to sweep to fixation, right? In that population, because we're imposing stringent selection in the body. Now in your colon, while this infection is live and active, I told you that in a previous lecture about how basically the population of salmonella explodes in your colon, leading to essentially watery diarrhea, is that now we're talking about a very large population, right? And so beneficial mutations arising and sweeping to fixation in your colon are much, much harder for that to happen for salmonella. Okay. Are there any questions so far? So I realize this is kind of meta in terms of this discussion, I suppose. And also mode of reproduction. So this is a big driver in terms of how evolution proceeds in populations. So if you are a sexually reproducing organism, what this ultimately does is it favors greater genetic diversity and phenotypic diversity, right? So there are a lot of different alleles in a population, and if you basically have to have sexual reproduction as part of forming new organisms, is that you have this sort of red and blue organism that come together and mate, and they ultimately produce a whole bunch of offspring that have wildly variable traits, right? So any of you with siblings, if you were to look at me and my sister, you might be able to squint and say like, yeah, I think they might be, but I would argue we don't look at them. But which is good for them. Anyway, so what this does for evolution is that when we start talking about how selection works on standing variation, is that one of the forces that can change frequencies of alleles is natural selection. So I've kind of talked about these selective environments of your gut. So the pH is really, really low. The osmolarity is really, really high. And there's a bunch of stuff that is there to essentially just kill you with your pathogen. And you being able to survive that is one of these kind of classic examples of like survival of the fittest, right? So evolution can proceed very quickly when there's a lot of standing variation. Because essentially you might say, all right, everyone that kind of is like purple to red in this population, this means that you have a particular allele variant that allows you to like, I don't know, survive extraordinarily low pH. And so all of you get to survive, and unfortunately, everyone else perish. So now we're talking about this particular red allele that kind of has varying degrees of phenotypic display in this population. Now lets all of these individuals move on. You have a lot of standing genetic variation all the time. Now, asexual reproduction, or replication by cloning, is something that the prokaryotic world is doing all of the time in the absence of horizontal gene transfer. And so mutations arising are subject to mutation rates. And so there's no kind of mixing of alleles, and you're generating a whole bunch of offspring that have enormous variation, right? So you essentially are producing clones, and that the only way things get different in the absence of horizontal transfer is basically through mutations showing up, and those mutations being beneficial in some respect, and that these things confer selective advantage. And so you can have large genetic diversity, but you end up with much lower phenotypic diversity frequently because most mutations, as we're going to talk about, are neutral. They don't do anything. So you might have a slightly different version of this particular protein versus your sibling. The chances are that it just means nothing in the context of prokaryotes. And so evolution by natural selection often occurs much more slowly if we're talking about sort of reasonable amounts of selection compared to sexual reproduction versus asexual reproduction. And for much more complicated organisms, this is one of the arguments of why sexual reproduction is there's lots of explanation as to why sexual reproduction is an amazing innovation in terms of genetics. But for where we're going to take our discussion, we're talking about asexual reproduction. All right, so I've invoked this idea of selection. And selection is essentially sort of sorting of alleles based on their respective benefit in the environment. And you can think about this in terms of natural selection, where mutations resulting in greater fitness are favored. And so this means, let's say this particular allele variant, that this was really the best allele for a particular phenotypic trait. And as generations march on, what I will see is that I'll see that this particular trait conferred an advantage. And so this particular individual was able to replicate more times than others. And subsequent generations, we see that essentially this entire population becomes dominated by this one allelic variant. And so this is one of the classic signatures of natural selection. So at the other bookend of what can happen for why you end up with genetic variation in prokaryotic populations, you end up with sort of fixation of alleles that do nothing, is a concept known as genetic drift. And so mutations of similar fitness can emerge simultaneously, show up, with one mutation rising to high frequency simply by chance. And so this is just, luck of the draw. There's just this one particular mutation that just floated around, had no particular benefit, but just eventually fixed in the population. And so there's some interesting arguments out there about why this happens. And one of the explanations is that the alleles that are showing up and just drifting around, we say drifting in the population, that they're just existing at some frequency, is that ultimately they end up hitchhiking on a really beneficial mutation and sweep to fixation along with that beneficial mutation. And that's why we see some of what we do, at least in the large population. So there, any questions so far? Is this super foreign for everybody? It was when I heard it. All right. So beneficial mutations.
So real quick, so
Yeah.
genetic drift, are the mutations, do they have to be close, or is it literally just related, or?
But like they can just be completely unrelated. So you might have, you know, mutation in gene A, mutation in gene B, mutation in gene C, all kind of existing in this population. All three are completely neutral. They don't confer a cost or a benefit to the organism. But let's say, you know, we're all trucking along. We're the prokaryotes were, you know, equally fecund, and that, you know, there are two daughter cells that emerge through binary fission. Everybody's growing at the same rate, but eventually, you know, you pick up a mutation in gene Z, which makes you grow this much faster than us two. Now your mutation in gene A, that's completely neutral, gets to hitchhike along with the benefit on the same genetic background as gene Z. That mutation. Yep. So I've invoked this idea of there being beneficial mutations. So one of the things that we know about mutations is that like in terms of thinking about what's going on with the emergence of mutations and the generation of standing variation in a population, is that mutations can be thought of as being drawn from drawn randomly from a nearly infinite possible mutations. So, you know, if there's 10 million base pairs in a genome, there's 4 possible mutations, or in terms of nucleotide change that can happen at each one of those sites. Now the number of possible mutations that can exist is astronomical, right? So we can sort of approximate this as being infinite in size. And so each mutation is essentially randomly drawn out of this pot. Now the thing that is interesting about this infinite number of possible mutations is that they don't all confer the same costs or benefits to the organism acquiring, right? And so what we can sort of conceptually think about with mutations is that they that they have defined fitness effects for that organism in a particular environment. And so if we want to conceptually think about this, is that we can start to think about this in terms of the distribution of what these fitness effects look like for an organism in terms of their mutations. So what we can do is, and this data is derived from from a viral model. And so viruses, because their genomes are very small, and because we can sort of rapidly assess what the fitness costs were for this virus in terms of their fecundity, the infectivity, all sorts of things, that, and this so far seems to be generally true for living things as well, is that if we plot the number of mutations, the frequency of particular mutations, if we took every single mutation that we could possibly make in an organism and ascribed and determined its relative fitness effect, so did that negatively impact an organism, which in the prokaryotic world would mean that it made you grow a little bit slower, made your cell division happen a little bit slower, for example, or was it beneficial in the sense that it made you much more positive, in that you grew faster, you divided faster, and so you were more fit than your competitors in an environment. If I were able to place every single mutation possible onto its relative fitness effect on this sort of x-axis here, and then I were to then look at the frequency of all of these different fitness effects of these mutations, what I can do then is develop what is known as this distribution of fitness effects. So there's an infinite possible universe of mutations that can happen, and then there are what are the effects of those mutations. And so this particular example is from a virus. There's lots and lots of other examples now for much more complicated living things. And they all follow this general interesting trend, which is that if we center around 0 and ask, what's going on with mutations that have 0 fitness effect, is that by and large, in this case for this particular virus, almost 50% of mutations have absolutely no effect on this virus, right? And so if we were to say, what does this estimate look like for us? It's much, 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 much higher. The vast majority of genetic differences that we have absolutely no effect on our relative fitness compared to each other. Now, what's also interesting is this far negative tail of this distribution, is that there are an awful lot of really bad mutations out there too. And similarly, if we were to just look at the general shape of this distribution, is there a lot more stuff more than 0 or less than 0? Less than 0, right? So that means that if we're going to sort of bias anything, is that the vast majority of mutations that are out there are bad. And so if we were to go back to our original sort of number crunching and ask, why the heck don't we see lots and lots of mutations in standing bacterial populations? It's because the vast majority of them have negative fitness effects. So as these organisms are growing and dividing and mutations show up, chances are that mutation is not beneficial, right? So they're going to be quickly culled from that population, which is one of the factors of why we don't see a lot of standing variation in those populations. And what's also important to note is that this distribution of fitness effects is highly dependent on environment, right? So a mutation in one environment might be really detrimental. So it might make you awful at growing in one particular environment. But in a separate environment, it might make you a total badass, right? And so one of the classic examples of this are target site mutations for antibiotics, right? So if I took E. coli in my lab and I pounded it with beta-lactam antibiotics, I would likely end up selecting for mutations in the enzyme transpeptidase. So transpeptidase is important for forming cross-links in peptidoglycan. Now, if I have a high concentration of some beta-lactam antibiotic, penicillin, in my growth media, that mutation is gangbusters in transpeptidase. It is going to make sure that organism doesn't die. Now, if I take that now penicillin-resistant transpeptidase E. coli and say, how well do you fare against wild-type E. coli? It's going to get its butt kicked, right? Because in the absence of antibiotics, now the efficiency of that transpeptidase reaction is much lower. So it can't build cell wall as fast as its non-mutated sort of ancestor. And so in one environment, this mutation was way out here, that it had an enormous positive fitness effect. Whereas in a sort of non-selective environment, now all of a sudden the relative fitness effect of that mutation is terrible. So it's highly negative. So what does this all mean? It means that evolution is both a consequence. Evolution and fixation of beneficial alleles in a population are both highly dependent on how big that population size is, what's the mutation rate, and also, importantly, what the heck is going on in the environment, right? Because all of these things are intertwined. So, are there any questions yet? Yes?
So on that graph, is that saying, since fitness has to do with survivability and reproducibility, is that saying that 0 has 0 effect on survivability and reproducibility, and -1 actually kills the cell?
Yes. In this particular, the way they scaled this data, is that, yes, like -1 was essentially like you're dead. You're no longer a viable virus. And so, am I answering your question?
Yeah.
Okay. Yes.
This is probably only mildly related, but I'm curious. So sometimes in like popular science media, you'll read about like old bacteria coming back again, like with the glaciers melting, something like that. But aren't we, evolutionarily speaking, aren't we more at risk dealing with pathogens that like invade the human population all the time and are growing towards becoming more fit within a human host than we are with some random bacteria that's making a reappearance? Like, wouldn't you say?
Arguably, yes. But, I mean, so us on the sort of grand scale of biological evolution, we're pretty short-lived creatures. So, frozen in the ice somewhere, there's some, like ultimate zoonotic pathogen. So when I say zoonotic, it means it can affect a lot of different hosts. Pathogen that's buried in the ice somewhere, maybe. But things that we see now are that host specialization is a major driver in kind of pathogens becoming better and better and better at interacting with us and the host. But zoonotic events, so these emergent pathogen events, are also really dangerous. So I think your intuition should.
I was just curious, like, are they trying to scare us, or like, should we be more concerned about people?
You should be more concerned.
Yeah, that's like.
Yeah. Yeah. So this all leads to, I think, an interesting set of observations that a lot of this is born out of experimental evolution in the lab, and the applications are really interesting in helping us interpret what happens with pathogens and provide some explanations for what we see in terms of the epidemiology of pathogens when we're sort of looking at a much larger scale. Why is this salmonella that infected these people in 2017 in Florida different from the same salmonella, arguably, that infected people in New York City, for example, this year. So anyway, this all comes down to what does all of everything that I kind of told you actually start to look like visually, right? And so I'm going to take you through 3 different regimes, as we call them, because we can think about how strong is selection. It's essentially like you are going to live or you're going to die type of selection, or is it sort of like, it'd be nice if you did this, but it's not required. But it'd be really cool if you kind of do this extra thing. And so that we can contrast, sort of bookend this and say there's very strong selection or there's very weak selection. And we can think about essentially how frequently are mutations showing up and how good are those mutations in terms of how beneficial are those mutations. So the first regime is this idea that there is strong selection, there's weak mutations that are out there that are beneficial in this particular environment. And so this pool of beneficial mutations under these conditions can be thought of as limited. And so the shape of that BFE is that there are very few beneficial mutations out there for you. And so what this means is that evolutionary events end up being strongly periodic. And I'll kind of give you a visual representation of this in just a second. But what this means is that you're essentially, as a population, always waiting for that next big innovation, right? And so it's essentially thinking about this like the world of in the world of cellular phones if there was only the iPhone, right? Which is almost true. But we'll just say that there's only the iPhone, and there's a limited number of, beneficial, incremental advances that can happen to this phone that are sufficiently beneficial that they will sweep through the Apple user population, right? And so essentially, you're always waiting for the next best phone, and that what ultimately happens is that Apple releases said phone twice a year, and so this becomes strongly periodic. So like, all right, every 6 months, that's when that next best Apple phone is going to come out. And this is what happens when there's strong selection for beneficial mutations, but the supply of those beneficial mutations is limited, right? So weak selection and strong mutation. So this means that beneficial mutations, they're very common, right? But there's very little selective pressure for any one of these genotypes to be the best beneficial mutation, right? So we kind of think about this in terms of, well, there's the world of cell phones, and this argument was a little better some years ago. But even now, if we say that there is the Apple and there's Samsung, and if we were kind of take a very global perspective, there's Huawei and a couple of other cell phone manufacturers, and that they're all doing cool things. They're all trying to make the best darn phone they can. And in the absence of any human preference, that that they can stably maintain themselves in this particular marketplace, right? Now, there's also this regime of strong selection and strong mutations. So this is where beneficial mutations, they're very common, and selective pressures are high. And so this is basically how I would argue, how the cell phone market actually played out, right? So this might have been like 2005, and this is 2018, right? And so what this does is it leads to strong competition between beneficial mutations. So these beneficial mutations arise, and the frequency that they show up is that they're temporally overlapping each other. And you end up with something that's known as clonal interference. And so this will visually make sense in the next slide. So here is sort of the one cell phone manufacturer world. And we can think about these in terms of what I'm showing you here is the abundance in population versus evolutionary time. And the way that I'm graphically representing this is in what's known as a Fisher-Miller plot. And what this is getting at is that the sort of proportion of what's in between these lines is the frequency, the relative frequency of, say, AB versus big A, little b versus little a, b, right? So if I were to say, at this particular evolutionary time, maybe 30% of the population is big A, little b, versus little a, little b, right? So this is, kind of think of, this is, does anyone know what the Apple, I'm not a really Apple person. What are the two things? There's like one around Christmas, right? Isn't that one of the?
September and November.
September and
November.
November for one of the releases?
Yeah, it's September and November is when they release it.
Okay. And that's for like, I thought they did it twice a year. There's like
yeah, those are the two releases. iPhones come out in September. Everything else comes out in November.
Just like iPads and
Yeah, like anything else comes out.
I'm just talking about the phone. So there was like
September
12th.
September, and then isn't there like one in June? It's like when that like the S version of everything comes out.
Oh, that's the year after.
Is that the year after?
But it's still the same.
Yeah, they only do phones once a year.
The iPhone one though.
Okay.
So the Note 9 got.
Okay.
Like, yeah.
All right.
Yeah.
So we'll say every year, right? So this is like, what is the iPhone up? Is like X something.
That's
XS. All right. So clearly we're on the verge of XI or something, right? So we're going to be 11 here soon, and that new phone is going to sweep through the user market to fixation, right? So at some point in time, every Apple iPhone owner is going to eventually own this thing. And the next one shows up, and we see this periodic event where, you know, these things, this new beneficial mutation, it's competing against the old organism, but essentially it eventually is going to win through its just there's beneficial allele, but and there's strong selection, but the sort of pot of beneficial mutations is not big, right? Now, on the other end, when I was talking about weak selection and strong mutations, is that there's a lot of beneficial mutations out there, but selection is weak. And this is what this ends up looking like, and that you can end up with multiple variants of, say, big A, so you have big A prime, big A star, and that these all kind of showed up relatively close to each other before any one of these big A mutations fixed in the population, right? And so essentially these things are not really duking it out because there's never any massive benefit of one of these versus the old A. It's just they are beneficial, but with respect to each other, they're all relatively equally beneficial. And so we end up with a massive amount of standing variation in this particular gene in terms of different alleles that you might see in a population. Now, this intermediate realm, the strong selection, strong mutation, is that strong mutations are showing up very rapidly. Whereas highly beneficial mutations are showing up very rapidly, and that there's also strong selection at play. So as soon as these things show up, they're on their way to sweeping to fixation. And this is sort of this, you might think of this as like, you know, this sort of 6-month alternating like iPhone, Galaxy, iPhone, Galaxy. But they're essentially competing for the same resources, and that ultimately, you know, arguably Apple was going to win this fight in the long run. But there's a good long stretch of time that these two alleles are coexisting in this population, and that they are essentially just duking it out, right? And so you end up with this sort of spectrum of outcomes, and that two of them, when there's strong selection, there's inevitably always going to be a winner, right? It's just when you're actually looking for that winner. So if I were to say it's mutation limited, you know, I would frequently see that there are periods of time where there's just 100% of something, and then if I were to kind of come back later, I would see this something new show up and sweep to fixation. Now, in this realm, if I were to take a snapshot, it might look an awful lot like this right here. But then if I came back later, I would see that, okay, there's something that clearly mopped the floor with everybody. Now out here, this is where you're basically going to see a lot of standing variation all the time. So these are three really important regimes in terms of evolution, evolutionary processes for asexual populations. And at different points in a pathogen's lifestyle, you know, what it's doing, where it's growing, all of these things can be at play, right? So what we see in for pathogens is that the host environment imposes strong selection, right? There's not really a weakly selective environment. And what it comes down to in terms of for a particular pathogen and its evolution is, are mutations strongly or weakly beneficial, and how quickly can they arrive, right? So what's out there in terms of beneficial mutations in that distribution of fitness effects. And so what that distribution of fitness effects for beneficial mutations looks like is a major driver in the success of a pathogen in a host, especially a new host, right? And so getting back to this question here, of like as you're becoming better and better and better at interacting with a specific host, you know, these are these processes that are driving what's happening. Now, the thing that can complicate this, and does, is horizontal gene transfer can operate as an interesting analog to sexual reproduction, and that it allows for generating great massive diversity in genotype and phenotype, right? So I've given you these examples in previous lectures of basically like you acquired this thing and now you're just a total badass, right? But, you were commensal and you had to fight with everybody over everything. Now you acquired this type 3 secretion system, you can invade host cells, and now, you've got the catbird seat in terms of, like, there's no one I have to compete with anymore. And unfortunately, now I'm pathogen. So, that, and I wanted to ask a question. So go ahead and put your pickers out. And here is one question for today. And this is that you have isolated a sample of sputum from a patient with severe bacterial pneumonia that has been determined to be caused by a bug, Klebsiella pneumoniae. I don't think any of you guys chose Klebsiella. So this patient has undergone several rounds of treatment with levofloxacin. This is a fluoroquinolone drug. Fluoroquinolones inhibit DNA gyrase. And so from serial treatments with this particular antibiotic, you know that this patient has undergone. You decide to do genotyping on a target of levofloxacin gyrase, so DNA gyrase gene. And in and amongst these 50 isolates that you have from this patient's basically kind of gross lung butter, if you will, is that there are 4 distinct populations of Klebsiella pneumoniae present. And this is just determined based on standing variation in this K pneumonia population. What I want you to think about, this means selection on the drug target gyrase was likely blank with respect to available dimethyl conditions.
Which is what?
Lung butter is the worst. You did.
All right, so I'm gonna go ahead and open up the window.
Yeah. That's good. Damn.
All right, so another 20 seconds or so.
I know,
5 seconds. All right. Whatever hold you is. All right. Okay. So, ooh, a good split. Nice. Does anyone want to make their argument for why it's strong? Okay, well, just that there are like clearly four standing groups of Klebsiella. Is that?
I think 3.
Okay, we got one for weak. Why?
Because if there's 4 populations, if it was strong, there'd only be one population, basically. Choose one above all the rest of them, and that one would survive, and all the rest of them would kind of die out. But if it's weak,
Then you can have standing variation.
Yeah.
Okay. What about antibiotic selection?
I think the strongest environment early and strong selection.
Right.
With exposure.
Okay. The answer is there's not enough data, right? Because if we go back and look at this, is that we're taking one snapshot, right? So we took one snapshot in time, and I said, okay, right there, I would see there are 3, right? And that would be a case that there is strong selection. And if I took one snapshot here, I would say, oh man, there are 4 different alleles in this same, 4 different types of mutations in this gyrate gene. And I would say, all right, well, it could be weak selection, right? And so what ultimately you need to be able to say anything about any of this is sampling across time, right? So taking something that you think is an endpoint snapshot of like this is how this happened. Because I could make the same argument here. If I only measured here and here, I could say, oh yeah, look, it looks like periodic selection, right? But that's not what happened. And so this is what has confounded sort of the study of evolution greatly, because getting sort of snapshots through evolutionary time, it's not easy, right? Especially if we're going to talk about human evolution, that's darn near impossible. Okay? So, in the microbial world, why we learn so much about evolutionary processes from microorganisms is because we can do this. So in my lab, I can perform these sorts of experiments and take whatever sort of time sampling I want and make these diagrams. I can understand an awful lot about what's going on with evolutionary processes in different environments. And so one of the big kind of challenges is, what's actually going on inside of a host, right? So we assume selection must be really strong, and so it must look like, what this clonal interference or mutation-limited types of environments may be true, or it may well be that that there is fairly weak selection after sort of the initial beneficial things have already happened. Now you're just talking about like kind of quibbling over is it like, well, is it 5% better or 1% better, not 70 to 80% better, right? So it all depends on that. Okay. So mechanisms of molecular evolution. So hopefully I'm not giving any sort of giant quantum leaps in innovation and thinking here, and that these are things that you've seen in a genetics course, and that different types of mutations can arise. These are things that are happening in the absence of horizontal transfer. You have single nucleotide polymorphisms, and this is what can lead to sort of very subtle changes in protein, amino acid sequence in a protein. And you have basically like synonymous or non-synonymous mutations. And the sort of ratio of synonymous to non-synonymous mutations in a protein coding gene can infer and tell you something about whether or not a particular type of mutation has been positively selected for an environment. And there's all kinds of interesting ideas that people have about this and inferring things about evolution of all sorts of things in different organisms. In prokaryotes, it kind of works out a little bit, but population sizes are really large, and they're sampling a lot of different types of mutations, so the sort of BNDS ratio is kind of doesn't tell you too much. Deletions, insertions, and so we talked a little bit about insertions, right? So you can have basically IOS elements move around. You can also have insertions of just sort of random stretches of DNA because of replication errors. Similarly, you can have deletions, you can have duplications, you have inversions and rearrangements. And so there's all kinds of things that can happen structurally to cause large mutations, sort of irrespective of horizontal transfer. And so SNPs, the single nucleotide polymorphisms, these can change promoter sequences, for example, increase, decrease expression of key genes. You can change amino acids in structural proteins, changing their function. There's a lot of things that can happen. Deletions typically quickly lead to inactivation of genes, so and frequently lead to genome reduction. Right? So this is kind of one of the cheap and easy ways that bacterial pathogens specialize, and I'll get to some of the examples in just a second. Now, one of the important things about duplication is that there's a really interesting hypothesis that's fairly well supported with an awful lot of data, and is actually, this is the backbone of comparative biology in terms of genomics. Being able to say like, well, I found this protein sequence, I did a database search, and it looks like a oxidoreductase. The whole sort of backdrop of why we can do that is based on this idea that duplications serve as a template for functional divergence. So if a gene duplicates itself, and you have two redundant copies of the same enzyme, now, depending on selective pressures, one can start to acquire mutations and become less functional in one particular activity than the original gene, and that this one can sort of evolve or can be selected to have new function, right? And so this duplication and divergence idea is the most parsimonious, and we talk about this in kind of evolutionary terms. Parsimony is sort of like, so what this, if I kind of look this up in the dictionary, it's talking about being greedy, right? And then it's the simplest or most direct explanation for something that happens biologically when we talk about parsimony in a biological context. And that this is the most direct explanation that we have for homologous enzymes and structural proteins that we observe in nature. So how do these things emerge? So that there once upon a time was some oxidoreductase, and that through duplication and divergence of all of these things, we end up with the massive diversity of oxidoreductases that we know, right? And so this draws a lot of support from sort of phylogenetics that we do to describe organismal divergence and speciation, that these same processes appear to be happening at gene level in terms of duplication and divergence and function. And one of the most remarkable is the evolution of the bacterial flagellum. And so when we actually look at the flagellum and look at the injectosome, which is this type 3 secretion system that we've spent a lot of time talking about in this class, is that structurally they're actually fairly analogous. and that the evolutionary origin of this type 3 secretion system, this needle complex, was actually this guy, right? And so there's actually an interesting amount of functional information that leads us to this conclusion, and that we can knock out some of these flagellar genes, say this protein FlyH. I can make a knockout of FlyH in E. coli and take this SCTL gene from Salmonella, for example, and I can complement this function in E. coli with this protein, right? So I mean, I have to produce an awful lot of it, but E. coli will swim again, given this particular protein. And so when we begin to compare at the amino acid sequence a large number of these proteins, is that there are functional equivalents between these two structures, and essentially there are a number of things that were lost along the way to make this injectosome. But where it began its molecular life was this bacterial flagellum. This bacterial flagellum, if we sort of tear apart its component parts, is that parts of this look like DNA replication machinery. Parts of this look like F1, F0 ATP synthase, so on and so forth. And so there's kind of a whole cottage industry now with the amount of structural information, with the amount of sort of DNA sequencing and protein sequence information we have to essentially reconstruct the origins of a lot of these extant molecular structures that we now observe in the prokaryotic world, for example, and where the hell did they come from? And the origins are interesting. So this classic example of this injectosome emerging from the flagellum. And so one of the interesting backdrops for this particular system is that for a very long time, this was kind of one of the sort of hallmark centerpieces for this intelligent design movement, which basically saying that these structures are irreducibly complex, which is wholeheartedly not true, right? That there is a very clear evolutionary origin for these components from this one. So much so that they're functionally compatible with each other in many cases. So, all right. So gene duplication and divergence is one of the massive ways that you get new sort of structures from old structures. And so what do signatures of evolution look like in pathogens? But before we do that, I have another question. Okay. So this one is the following. So you're working with a particular pathogen, Coxiella burnetii. I don't think anyone's working with this semester, but it's the causative agent of Q fever. And so it's a particularly nasty pathogen if you get it, and it's typically associated with sheep. And so you have identified a cluster of genes involved in pathogenesis by a transposon sequencing experiment in a mouse. And genetic knockouts of these genes results in decreased delivery of effector proteins into host cells and tissue culture. So Coxiella burnetii is an obligate intracellular pathogen. It has to live its life inside of host cells. And so when you look at many of these genes, you see that many of these genes look similar to genes involved in construction of these E. coli F sex pills. So what secretion system would you expect this?
What about my green?
All right, so another 25, 30 seconds. Final chance. Ooh, under the wire. All right. So, a good number of D's and a smattering of B's, C's, and E's. All right. Whoop. Okay, so the answer is indeed D. It's a type 4 secretion system. So this F Saxpilus, once upon a time, I described this last time that it's where you see type 4 secretion systems frequently kind of come up now is in sort of mobilization of DNA, transfer of DNA between bacteria. Well, it appears its sort of molecular evolutionary origin was actually transfer of proteins once upon a time. And many type 4 secretion systems that exist still in many pathogens, still this is what they do. They deliver proteins and not DNA. So, let's down to the wire. So, I want to kind of finish a thought today, is that all of this boils down to some bigger observations that we make about pathogens, because all the sort of things that I took you through are things that we like to try and infer. So I can do things in the lab that sort of resemble what happens in a host. I can put things into a mouse. I can do all sorts of things. But end of the day, there is a certain amount of contrivedness to sort of seeing all of these different kind of processes and saying mutation rates and population size and all of these things. And then there's what we actually observe out in the real world. And so what actually happens in most infections is to keep in mind that successful infections are by very small subsets of pathogens, right? And so it requires a reasonable number of pathogens to show up. But those populations that are actually successful are very small. And what this results in is that highly similar clones propagate in the host, right? That what we clearly observed, if we look at sort of why was this salmonella outbreak in Florida so different than New York, is that these were different selective events that happened. And so that dissemination of this particular successful infection event to other individuals in the human population, for example, result in proliferation of this clone. And what this is important is that essentially what we see, why we see so many different serovars of pathogens and all these sort of things isolated from different places that differ so wildly from each other, is that essentially we have these sort of selective events. We're selecting for, yeah, all right, this particular salmonella is the winner this time, and we have a population boom of this one particular salmonella, and that essentially this is clonal expansion. So those of you that have taken an immunology class, this is kind of like what happens with healthy T cells, right? That they get activated, proliferate, sticker B cells, they proliferate, and then you end up with like a whole bunch of this, right? This is clonal expansion of this one version of salmonella. And what this does for us in terms of epidemiology is it makes outbreaks traceable, right? So we know we have this operating assumption that we have clonal expansion, which generally appears to be true. which means we can track things back to where particular infection events happen and potentially when they happen. So, I believe our discussion where.
But you did not get six.
Okay. Most importantly, just get together.
She's standing there.
I just